Good morning, everybody. My name is Professor Suzuki, and this is Brain and Behavior. It's a MAP course that satisfies the Natural Science 2 requirement. So this class focuses on the discipline of neuroscience, trying to understand how the brain allows us to see, feel, sense, remember, and do all the things that we do and all the things that make us who we are. I want to make a distinction between neuroscience and psychology. So psychology is really the study of behavior, very fascinating study. But we're going a little bit deeper than that, trying to understand what exactly our brain is doing. What are the connections and the synapses and the neurons and the circuits doing that allows us to do all the amazing things that we do. So let me just get a quick overview of the class. How many freshmen do I have in here? Okay, great. Well, welcome. Sophomores, juniors, and seniors. Great. Okay. Happy to have you all. Let me just give you a quick overview of the kinds of things that we're going to be learning about. We're going to start with the basics, some basic brain neuroanatomy. What parts of the brain are there? Where are they relative to each other? What is the three-dimensional organization of different structures in the brain? We've developed for the first time this year a lab that I think is going to be very useful and exciting for you. It is a sheep brain, human brain, digital atlas that you're going to actually create on your own in LabCourse. You're going to be able to take away from that lab your own personalized digital atlas of a comparative sheep brain, human brain atlas so that you can remember all the different brain structures that you used. We're going to be talking about brain structures. Then in the first chunk of the class, there's kind of three parts of the class. In the first chunk, we'll really dive into the building blocks of the brain. Now, the brain really only has two types of cells in it. The entire brain is made up of two types of cells. Does anybody know what kinds of cells they are? What are the names of these two kinds of cells? Anybody? Wild guess. Neurons? Second type? Starts with the G? Glia. Okay. The whole brain is made up of only neurons and glia, just two types of cells. And of course, there's different kinds of neurons, different kinds of glia, but really it's only two main parts. And our goal is trying to figure out how they're put together in all these different systems. We're going to be talking about neurons, glia, and their interactions, their communications, which are synaptic communications. They communicate through a combination of electrical and chemical synapses. A huge chunk of the kind of conceptual work that we're going to be doing in this first third of the class will be understanding those synaptic communications. You people that came in, Eric's going to give you a syllabus over here. We're also going to be talking about neurotransmitters. In the second part of the course, we'll move on to sensory systems. So how do we see? How do we hear? How do we feel? How do we smell? We're going to be talking about the general principles of organization that are similar across all the sensory systems. And then we're going to move on to the motor system. So once the sensory information comes in, you want to do something about it. You want to act in the world. How is that motor system organized? And then finally, the third part of the class, we will focus on higher cognitive functions. I'm talking about learning, memory, emotion, language. We're going to end with one of my favorite lectures, which is called The Neurobiology of Love. So what do we know about social attachments and the parts of the brain important for social attachments? So beyond that, those are kind of the basic bare bones topics that we're going to go over. I do have three major goals for this class. The first goal is to give you a little bit of inspiration. We're going to be talking about Nobel Prize winning science here. And in many different areas. There's Nobel Prizes in the study of the neuroanatomy of the brain, Nobel Prizes in the study of the visual system, a Nobel Prize for the study of the molecular biology of learning and memory. My goal is that something across this entire semester is really going to inspire you. Maybe it's not cells and neurons. Maybe it's really language centers, hemispheric specializations. But I invite you to be open to that inspiration through the class. Second, one of my main goals for this class, and this is the second time I've taught this, a big class like this, is much more interaction. So in a lot of these larger classes, it's all about the professor standing up here and talking, and you're just sitting there kind of passively. But we all know that the best way to learn is to actually get involved, get interactive. And so to try and facilitate that, the TAs and I have come up with 3 interactive projects for us to do, each one in each of the thirds of the classes. You'll be coming up here, lots of interaction, lots of opportunity to get involved. I invite you and encourage you to ask questions during class. Now, this is a big room. I may not be looking over here when you're asking. Be a little persistent. Wave your hand. I will always be happy to answer any questions during class. So please, please do that. And also, I also want to encourage you all. This is a big class. There's a lot of information to learn. And particularly if you just started here, if you're freshmen or sophomores, you may not know a lot of people in the class. I encourage you to get to know the people in the lecture, but particularly in your lab sections. Form study groups. Use your fellow students to help learn this material. And to kind of get this ball rolling, what I want you to do is turn around and introduce yourself to 2 people. Tell them your name and tell them what lab section you're in or you want to be. Please do that right now. Okay, great. Hopefully you've made 2 new friends. You will follow them through the class. You will help them out. You will form study groups. You will come to me as groups and ask me whatever questions that you want to clarify anything you want. This is the best way we know how to learn. Get interactive. Get involved in this topic matter. And the best way to do that is meet your fellow students. Okay, so that's the interaction goal. The third goal is appreciation, and this is a huge one. I really focused on this last year as well. Appreciation, by appreciation, I mean I want you guys to gain an appreciation of all the different ways you see neuroscience in your lives. It's all over the news. It's all over the New York Times. It's all over different plays that you see here in New York. And so one of my goals is to really open your eyes to all the neuroscience around you. And to help you with that, we have 2 different homeworks that I'll talk about in a little while. That will encourage this. A media report where I'll ask you to go out and read something in the newspaper and write a little report associated with neuroscience, and then a movie project where we'll ask you to watch one of a wide range of movies that's associated with neuroscience or neurology, and talk about how accurate those are relative to what you've been learning in class. Okay? So inspiration, interaction, and appreciation are my overall goals of this class. Speaking of interactions, I just want to mention that this class is being filmed for what's called the Open Education Project. So there is a camera back there, and there are operators, and they're filming me. They're filming the lecture part of the class. We'll have interactive exercises up during the course of the class, and those won't be filmed. Okay? Anytime a student is involved, there's no kind of conflict of interest, there's no privacy issues, you will not be filmed. The filming is about the lectures that I'm giving. And the only thing is that I wanted to do this, I just want to let you know I wanted to do this because I think it's important to open up this kind of information for as many people as possible. So there's 150 people here, but this is going to be on the web. Anybody can download these images, and it's a great opportunity to spread this information for anybody that's interested. And 2, it kind of forces me and encourages me and inspires me to be as clear and concise as I can, because I know it's not just you guys that are watching, but it's anybody that can look at these lectures. So you won't notice the camera. You won't be on camera, and the only thing that I'll do slightly differently is anytime a question is asked, I will repeat that question so the microphone can pick it up, but also so everybody else can hear the answer. Okay, so let me tell you just a little bit about myself. So I'm a professor of neuroscience and psychology. My areas of research are the neurophysiology of memory. I, for the last 15 years, have studied the patterns of neural activity that allow us to form and retain new long-term memories for facts and events. And more recently, I've become interested in the effects of exercise on learning, memory, and cognition in humans. As a subproduct of that, I'm actually also a certified group fitness instructor, and I teach a free exercise class every Sunday at the Palladium, at the third floor multipurpose room. It's a high-impact, high aerobics class. Every Sunday throughout the semester, you are welcome to come. I will tell you all about the exercise research that we're doing, and you'll get a great workout. So there's a sign-up sheet up here if you want information about that. So the other thing that I'm involved with is I've co-founded an organization on campus called Empowering Women in Science. It's actually both for men and women. It's really leadership training. I run an explorations dorm floor also at the Palladium called Empowering Women in Science, where we give free leadership training seminars. We also run a variety of different programs where we bring women scientists in, bring people, scientists from the community in to talk, and I will be telling you about that. If you're interested in learning more about that, you can sign up on the sheets in front. And just to give you a little bit of a heads up, one of the programs we're working on for the fall for Empowering Women in Science is a program where we're going to try and bring the actor James Franco, and he's actually a professor here at NYU, and he just had a movie out called Rise of the Planet of the Apes. And so the program we're trying to put together is called Understanding the Monkey Mind, in conversation, Wendy Suzuki in conversation with James Franco. So I will let you know about that, and hopefully that will be something that will happen in the fall. Other courses that I teach are a course called Can Exercise Change Your Brain? A course where we go over all the neuroscience underlying how exercise can change the brain's anatomy, physiology, and function. And at the same time, you get an hour workout during the class as well. It's very popular. And I also teach a class called the Neurophysiology of Memory, which really is based on all the work that I've done in understanding the brain basis of memory. Okay, contact information. I encourage you all to go to my website, www.wendysuzuki.com. You can read all about the research that we're doing, see a little bit more about the exercise studies. I'm on Twitter. I'm going to be tweeting for this class all about all the interesting neuroscience that I see in the newspaper, in the popular press, and this is going to be very valuable for your media project because you're all perfectly capable of going out, but I can give you some hints of different articles that I found particularly interesting. Our hashtag is BNBF 2011, Brain and Behavior Fall 2011. I also tweet about other things, but tweets particularly associated with this class, I will use that hashtag. My email address is here. I don't have set office hours because that usually doesn't work. You want to see me, email me, and we'll find a time anytime. So always open to meeting with any of the students in the class. Teaching assistants, we have 2 assigned teaching assistants right now. A third will be assigned at the end of the week. Eric Nyland, say hello, is one of our stellar teaching assistants. He will be running 2 of the labs. Which labs are you doing again?
The 2 afternoon Thursday ones.
2 afternoon Thursday ones. Oh, let me just tell you that Eric or myself or Sarah, who can't be here today, we don't have anything to do with the assignment of labs or or allowing you to be in the course. That is all done through the MAP office. So any of those kinds of questions go directly to the MAP office because we can't intervene for those kinds of things. The MAP office takes care of everything. Okay, a little bit about the lectures. As you know, the lectures for this class, Monday and Wednesdays, 11 to 12:15. Lectures are mandatory. As you'll see, we go over a lot of information. The book that we're using is Biological Psychology in the bookstore, 6th edition. And the reason why you need to come to class is that you don't need to know this whole book. Thank goodness, right? I'm going to go over select parts of the book, and the only way you're going to know what part we're going to be emphasizing is to come to class, okay? The other required text is our lab manual, also available at the bookstore. Okay, homework. There's going to be 3 homeworks that I'll assign in class. One of them is a media report, one of them is an experiment on the auditory system, and the third one is the movie project. We'll give you written instructions on exactly how to do that. The exams, there are 2 in-class midterm exams and 1 comprehensive final. Big red mark says no makeup exams. I don't care if you're sick. Makeup exams are not given, and what we do is we have the final exam count more. You don't want that to happen because the final exam is more difficult. It's comprehensive. Please try and make the midterms if you possibly can. Grading is very much based on the exams. 70% exams, 25% your labs, and 5% homework. All course information, all lectures are going to be posted on Blackboard. The lectures, the syllabus are all on Blackboard right now. And finally, please write this down today. The final for this class is Monday, December 19th, in this room, 10 to 11:50. Okay? I'm going to repeat that multiple times. You cannot miss the final. Make sure you're here on Monday, December 19th. It's great. It's at the very beginning of finals. So we get to get the final over with early in the finals period. Okay, lab sections, also mandatory. Let me just say, and Eric and the other TAs are going to emphasize, that we are going to be working with brain tissue. You're going to be doing a sheep brain dissection lab. You're going to be working with rats at the end of the semester in a rat behavior lab. These are mandatory. Squeamishness is not an excuse not to come to lab. And if you don't want to do it, then there's lots of other neuroscience or natural science MAP classes that you can take. These are mandatory. It's critical that you guys get a hands-on feel. The lab component really gets you into science and gets you kind of doing experiments on your own. So it's absolutely mandatory that you are that you participate in all these labs. Let me just mention that religious holidays coming up. There's 2 that will affect this class. One, September 29th and 30th. Some of you may not be present for the review that we're going to have for exam 1 during those lab sections. Never fear. For those of you who won't be here, you must inform your TA, and the TAs will organize an alternative review section that will be in a time that you can be here. The other conflict could be October 13th and 14th. This is a neuronal visualization lab. For that one, if you will be away because of the religious holiday, you must inform the TA before you miss the class, and that lab will not be counted if you're going to be gone for the religious holiday. Okay, so that's all the logistics. Let's get down to what we're here to study, which is the human brain. So this is a real human brain. It's actually really dense. If we have time at the end of the class, you can come up and throw on a pair of gloves and pick it up. But this is it. This is what we're going to be studying for the next 15 weeks. How this allows us to see, feel, hear, creates our own personalities. So let me just show you a few aspects of this brain. And actually, let me just tell you, talking about inspiration, so I'm asking you to be a little bit open to inspiration. The reason why I'm standing in front of you today is because when I was a freshman at UC Berkeley, I took a freshman seminar class. There were only 10 or 15 of us, with a professor. Her name is Marion Diamond. She's a neuroanatomist that has studied brain plasticity. And she pulled out a human brain just like this. And she said that what I'm holding is the most complex structure known to mankind. It's the only structure that can think about itself. Think about that for a second. Understanding the brain. We're so far away from understanding everything. But what we're going to try and do is give you a flavor for how we've approached it and how far we've come in our understanding of brain function. Okay, if this was my brain, it'd be sitting in my head like this. This is the frontal lobe, and my little eyes would be sitting right here, okay, looking out from right here. This is the frontal lobe, critical for our personality, critical for things like working memory, and critical for things like keeping track of items, okay? Let's go to the back of the brain here, way at the back of your head. This is called the occipital lobe. Occipital lobe is important for what? Anybody know what's here at the back of the brain? What function is mainly here? Vision, very good. So that's why when you hit the back of your head, you see stars. You're physically stimulating this part of the brain, and it's causing activation of your visual cortex, okay? If you damage this area of the brain, whatever, gunshot wound, accident, your eyes could be perfectly functioning, you will be blind. This is the part of your brain that allows you to see. Let's move up here. This is the parietal lobe, important for what's called visual spatial functions. People with damage to the parietal lobe have what's called neglect. So if you have right parietal damage, you will ignore everything on your left side. You will not eat anything on the left side of your plate. When asked to draw a clock, you will only draw the right side of the clock and not the left side of the clock. Even though you say, you have only drawn, the person will say, you've only drawn half the clock, you'll not be able to see that. You'll not be able to appreciate that. Visual spatial functions. And finally, this last lobe is the temporal lobe. Important for higher level vision and important for processing faces. We're going to be talking about that a little later in the class. And the last thing I want to show you is to flip this over. This is the cerebellum. Anybody know what the cerebellum does? Yes? Motor functions, excellent. Fine motor functions. So being able to learn fine motor functions, things like writing, things like sewing, are dependent on the cerebellum. Take a look here. What's the difference here? Yeah, so there's a huge difference in this brain. His or her left cerebellum is about twice as big as the right cerebellum. We all, any one of us in the classroom may have these kinds of asymmetries. In a normal brain, they're both the same size. I don't know whether this caused any problems, but there's lots of variations that you see in brain structure. But what I wanted to focus on is down here. This is the bottom part of the temporal lobe. And down here, right below this cortex, is a structure called the hippocampus. This is a structure that I've studied for the last 20 years, and it's critical for our ability to learn and retain new long-term memories for facts and events. Alzheimer's disease attacks the hippocampus first. And what is the first symptom of Alzheimer's disease? Memory loss. That because the hippocampus is being damaged. There are formations of what's called plaques and tangles, specifically in the hippocampus. What's devastating is that those plaques and tangles then slowly but steadily move out across and into these cortical areas. First in the temporal lobe, up to the frontal lobe, and finally back into the whole cortex. That's when you start seeing more and more severe dementia. Okay? The amygdala, important for emotion, is right next to the hippocampus. When that starts to get affected, then your emotional responses become abnormal. And finally, the endpoint of Alzheimer's disease is death, because those plaques and tangles end up innervating and infiltrating the parts of the brain in the brainstem, critical for breathing and heart rate. And without that, your system cannot function. So that is the location of the hippocampus. But just to give you a little bit of a flavor of what you're going to be focusing on, not in the first lab, but in the second and third labs, understanding the function of this structure. Okay. Okay, so what I like to do in these opening lectures is give you a little bit of an overview. But what I always do is try and choose a couple of examples that have been most striking to me about something that I've seen in neuroscience recently. Last year, there happened to be a huge number of articles in the New York Times about neuroscience, so I just went through those and just illustrated how much neuroscience is in the news. This year, it was 2 books that were really striking and interesting to me. And the first book is a book that was recently published called Portraits of the Mind, Visualizing the Brain from Antiquity to the 21st Century. It was written by, he's a graduate student at Columbia in neuroscience graduate school, but for many years has had a fascination with collecting neuroscience illustrations from the most beautiful illustrations, from the most kind of technologically advanced techniques we have today, all the way through antiquity. And he put them together in really a beautiful book. So this was a great example of, just to give you some portraits and to show you the beauty of the nervous system and how it's been illustrated over time. So we're going to start with some of the early pictures. This is just a quick overview, but anybody interested, I'd really recommend that you check this book out. Here is a picture, a reproduction of the oldest known drawing of the visual system from 1027. This drawing was made in Cairo. And what we're looking at here is, this is a picture of the eyes. Here's a little nose. And what we're looking at is the optic nerve coming up and crossing here at the optic chiasm. You're going to see all of that in the sheep brain dissection that we do in the second lab. So even as early as 1027, people were interested in understanding the neuroanatomy and how vision gets to the brain. Okay, now we're going to jump to the 19th century with the study that has become known as phrenology. Now, phrenology got a bad rap. During this time, people were learning more and more about the brain and starting to develop the hypothesis that maybe different parts of the brain were important for different functions. An early idea was that, well, the brain is involved in so many complex higher order functions, as probably all of it has to participate, so there's no localization of function. Some thought that in fact there were, and certain parts of the brain were important for certain functions, and just like a muscle, if you had very strong intelligence, then the intelligence part of your brain would start to bulge out. Maybe not so unusual or far-fetched an idea. Phrenologists took it one step further. They said, okay, if you have an intelligence part of the brain and it bulges out because you're so intelligent, then the skull is going to compensate. You're going to get a little bulge in that skull because your brain is bulging out. So therefore, if I feel the bumps on your brain and I correlate that to whether you're really intelligent or whether you have great motor skills or whether you are a great writer, then I'm going to be able to understand just from the bumps on the brain, on the skull, exactly how the brain function is organized. Well, they went a little bit too far because they subdivided the brain into way too many different parts, and in the end, the bumps on the brain were not very consistent. Were not consistent at all across people, though years and years of study was devoted to this. And here you see a skull where one of the German phrenologists has drawn on their rendition of what parts of the brain are important for, what parts of the skull are representing certain brain functions. So phrenologists, in the end, were not respected, and that fell off the map. But you see a remnant of phrenology today, and you're all familiar with it. Have you seen these drawings? This comes from phrenology, okay? And there's a little bit of truth in these kinds of renditions. And certainly, there are differences and maps made between the female. And I have to tell you, I spent a long time to find one that wasn't completely offensive. So this is a kind of cleaned up version of the male-female brain, but it also brings up something that we're going to be talking about, sex differences in the brain. So I have this brain right here. Who here thinks that it's a male or a female brain? Who thinks it's a female brain? Who thinks it's a male brain? Okay. Why do you think it's a male brain? You just, okay. Just participating. Thank you. That's good. That's good. The fact is that looking at the surface of the brain, you cannot tell anything. You can't even tell. You think, well, I know some friend, he has a huge head. Maybe he has, a really big brain. Everybody knows that the size of your head has nothing to do with how intelligent you are. Why? Because intelligence has to do with how your brain is connected, and that is not due to the size. You can't tell from the surface. What you can differentiate are the connections, the neurotransmitters that are working or not working, the receptors that are present or not present, and they're causing differences between male and female brain. We're going to be talking about what some of those differences are. Very interesting to start to look at that. That is a relatively new field. People have been interested in this for a long time. But we're going to be talking about some of those things that really make some of the physiological and some subtle anatomical differences that we know exist in the brain. You can't see them on the surface, but they do exist if you look microscopically. Okay, so let's jump back. 1875. Here we're jumping back to one of the first ways that we had to start to look at brain structure. This is a technique, it's a staining technique developed by a neuroanatomist named Camillo Golgi. He was Italian, and this is one of Golgi's beautiful drawings of the olfactory bulb using his technique. Now, let me just emphasize that he never saw this beautiful picture that he drew here. He compiled this picture from lots of different sections. In this stain, what you have to do is you have to use dead brain tissue, and then you have to process it in very, very caustic chemicals. And what happens is, every once in a while, you'll get a beautiful section with only one neuron labeled. But that neuron, the entire body of the neuron, the input parts, the dendrites, the cell body, and the output, the axons, will be entirely labeled. They still don't know why, but you can get those labeling in this section. So you can imagine that he labeled hundreds and hundreds of sections, and excuse me, he got certain sections that gave nice labeling, and he knew which structure he was staining, and so he put this together based on all of his different labeling. Very intensive kind of work to do, but that was the early way that people in the early 1900s were first able to start to understand how the brain was made up and what neurons really look like for the very first time. So Camillo Golgi was one of our Nobel Prize winners. He shared the Nobel Prize with this gentleman. Santiago Ramon y Cajal is known as the father of neuroscience. He was a brilliant neuroanatomist. He used the same stain that Camillo, he used the stain that Camillo Golgi developed, but he was able to pull out a very fragmented data, principles of organization that we now know are true today and that were confirmed with much more powerful techniques than he had at his disposal. He used the Golgi stain, his eyes, and a really rinky-dink early microscope. That's all he had. And this gives you a beautiful illustration. This is one of his illustrations, again, a compiled illustration. He never saw this. He developed this from lots and lots of different stains that he did of the retina, where you see the primary sensory neurons of the retina up here, the rods and the cones connecting to the two other layers in the retina, and finally going out. And so he saw this. He knew what the eyes were for. The eyes are for vision. And what he noticed is that the dendrites in this structure, and in general, tend to be of lesser extent. They extend out lesser amount than the axons that can travel a long, long distance. And he gleaned from that what is happening in this neuron, in these neurons, is probably a unidirectional flow of information. And he certainly saw this in the retina, where he knew that all the information was going in one direction, and all the neurons were kind of facing in that direction, with the dendrites on one end and the axons on the other hand, meeting the dendrites of the next cell towards the axons of the other. And so he developed this idea of the principle of directional specificity. Information always flows from the dendrites to the cell body to the axon. Now, the only way to confirm this is with physiology, which didn't come on online for many, many different years. And he was able to glean these just by looking at his neuroanatomical preparations, which is why he is still considered such a genius in the field of neuroscience. Okay, so we have this stain, and for many years, including today, people still use the Golgi stain to look at dead tissue and figure out what the connections are. But there's a problem. You can only study dead tissue, and the stain is only black. If you're lucky, you get one cell that's stained, and in other sections, you get a whole black mass because there's 30 cells or 100 cells that are stained, and there's no way to tell the difference because they're all black. You have to tell me there's a better technique than that. And for many, many years, in fact, up till about, I don't know, 8 years ago, the answer was no. That was one of the premier ways to look at the anatomy. But we come to a new era of neuroscience, and the new era starts with a lowly, what is this called? A jellyfish, sorry. The lowly jellyfish. So this is also a great example of what's called the value of what's called basic science. So basic science is curiosity, studying science for curiosity. I'm a basic neuroscientist. I study memory. Now, my studies are related to Alzheimer's disease and aging, where memory gets worse, but I don't study Alzheimer's disease and aging. I'm interested in how memory works. Neurobiologist was interested in how the jellyfish fluoresces, okay? And why is that important? Because this curiosity, you never know where this finding is going to go. This biochemist, who actually won the Nobel Prize for this, had no idea that he was going to develop the next best technique to the Golgi technique when he started studying bioluminescence, but he did. So he was interested in the proteins that allowed this jellyfish to fluoresce. And he turned out to find two different proteins, one that fluoresced blue and one that fluoresced green. And the green one would fluoresce green only in the presence of the blue one. Okay? The blue one had a name, but the one we're going to focus on is called green fluorescent protein. Okay? So the DNA sequence for green fluorescent protein, or GFP, as you'll hear it's called, was then determined. So we figured out what is the code of DNA that codes for green fluorescent protein in the DNA. And then another neuroscientist said, well, I'm going to play with that, and I'm going to actually insert green fluorescent protein into the DNA of a worm nervous system. And what happened is the worm nervous system lit up like a green Christmas tree. And you got, oh, sorry. So Shimomura was the biochemist that looked at, that first discovered green fluorescent protein. And Chalfie was the one that first put green fluorescent protein into the DNA of worms. And what he got were these worm neurons that were fluorescing green. Yes? What year was this? This was in the 80s, late 80s. Well, actually, sorry, it goes for a long time. It was in the 60s that Shimomura found the different kinds of fluorescent protein. And then in the 80s into the 90s, Chalfie put it into the worm. And so we got these beautiful images. So now we've moved from black stain to a beautiful green stain. But the cool thing is, with the technology of molecular biology, you could actually insert green fluorescent protein into an entire organism. And the main organism that we use in molecular biology in these genetic approaches is the mouse. And so you could actually make a green fluorescing mouse. This is not a joke. So the whole, it doesn't hurt the cells. I mean, it's completely innocuous. But these are mice where we're shining a blue light on it, and you can see which 3 mice have GFP inserted into all their cells. They're glowing green. Okay, so this is cool, and what it allows us to do is 2 things that we weren't able to do with the Golgi stain. One, we are able now to study live cells in the brain, And two, we're able, with molecular biology, to then insert GFP into specific kinds of cells in the brain. So we could study a whole class of cells at the same time. Much more powerful approach. But then you say, well, everything's green. So you still have the problem of if there's too many green cells at the same time, you still have a hard time visualizing. And that's where the third researcher who shared this Nobel Prize, all three of these up here, shared the Nobel Prize, Roger Chen, who started playing with green fluorescent protein and looking at the fluorescent proteins of not just this jellyfish, but other fluorescing jellyfishes to start to tweak those colors and get a whole series of different colors. So Chen kind of got the ball rolling, and others jumped in to really identify so many different colors, and they called it brainbow. Okay, so this is a real term. It's brainbow. And what they're able to do now is genetically go in. And unfortunately, what they can't do is say, OK, I want all cell A to be green. And then I want all cell B to be red and all cell C to be yellow. They get a random array of cells, but you're able to visualize many more cells together in circuits. And you can imagine that this is so important for the study of the brain, because there are hundreds of billions of cells in the brain. And imagine what the task of poor Ramón y Cajal and Camilo Golgi was by studying one cell at a time. Okay? What you really want to do is study circuits, and one particular circuit important for one thing, and not just the cells, but their synaptic connections. We're getting a little bit closer to this, but it's still not perfect. But I'll just show you a couple of the really beautifully stunning pictures that you get with this brainbow technique. This is brainbow applied to the cerebellum, ending in presynaptic what's called rosettes. And so you can see lots of different cells have been labeled, and here are their axons, and they're labeling these little puffs are the rosettes ending in the cerebellum. And here are axons of motor neurons that are all, again, colored with different colors of the brainbow, and you see all of their axons going down. This is a critical point of visualization. So there are cells, and you can see the cell bodies, but their axons could go for meters and meters. So I want you all to close your eyes for just a second. Close your eyes, be completely still, and everybody move your right toe. Still and moving your right toe. Okay, open your eyes. So here's what happened. Your left brain, your primary motor cortex is right about here. You had a whole bunch of neurons that represent the motor output of your right toe. They were firing like mad. Their axons went from here all the way down your spine, all the way down your leg, and they terminated in those muscles that were moving your right toe. That's a huge pathway to go. Imagine how long Shaquille O'Neal's axons for his right toe are. Now imagine a giraffe that is moving his right hoof. That's a motor neuron that's going from his tiny little giraffe brain. Sorry, no offense to the giraffes, but they're tiny. But he does have a motor cortex all the way down the neck, all the way down. So this gives us a way to visualize these very, very long pathways. Okay, and then I just want to end this part of the book with methods that I'm sure you've heard of, magnetic resonance imaging, MRI. It's been around for a while, but I just want to point out that it has been so valuable for the study of neuroscience now that MRI images, just structural MRI images, have become so precise in their resolution. This is the brain of a very famous patient named HM. Anybody heard of patient HM? Yes. What was he famous for? That's close. That's Phineas Gage. This is HM. Yes. Excellent, yes. He had anterograde amnesia. So when he was 27, he suffered, well, actually, from the time he was about 8 till he was 27, he suffered from very, very severe epilepsy. And a common treatment for epilepsy is removal of one hippocampus. I talked about the hippocampus here. What's the hippocampus important for? Memory. So this was before we understood how important the hippocampus was for memory. And his epilepsy was so severe that they decided to remove both hippocampi just to make sure that the epilepsy was alleviated. Well, they did alleviate his epilepsy, but from the moment that he woke up when he was 27 years old till he passed away just a couple of years ago when he was in his 80s, he had no ability to put any new information into his long-term memory. And for many years, so of course the operation was done in the 50s, so they had no way to image. And for a long time, it was thought that the aneurysm clips that they put in his brain during his surgery were ferromagnetic, so he couldn't be imaged using MRI. It was later discovered that, in fact, those clips are non-ferromagnetic. So we were able to get an amazing live picture of what his brain looked like while he was still alive. And this is an image from that. So on the left-hand side, you see images from HM's brain. So you can see these holes are where the damage was. And on the right-hand side are control brains. So you can see what the normal brain looks like. In the first top two slides, on the right, you see the amygdala, important for emotion, that's damaged in patient HM. HM was said to have a flat affect, probably due to the amygdala damage. Here, we're starting the hippocampus. Here, clearly damage to the hippocampus. And here, very tiny hippocampus left. So through the study of patient HM, who to this day is probably the most carefully studied neurological patient that existed, we have started to understand the organization of how memory works in the brain, focused on this key structure, the hippocampus. Now, we've gone a long way from these simple structural images to a new technique called diffusion MRI, which allows us to image, again, those effusive and very, very thin axons, those outputs of the neurons. And here we're looking at the axons in the cerebral cortex. And this is from a patient with damage to the thalamus and the midbrain. Anybody have a guess where that damage is shown on this slide? Where do you think the damage is? Where are you pointing?
Part where it's like
Up or down?
down.
Down? This, yeah. So this, exactly. So here, all these little terminals are little pieces of axons that are just damaged. They're here. This is the thalamus, and this is the midbrain. These are healthy axons, and then you can see exactly where that damage is. So it's a very precise way to look not just at where the cells are damaged, to get this precise image of where the fibers are damaged as well. Okay, so that gives you a little overview of some of the kind of pictorial representations of the history of neuroscience and where we are in kind of visualization functions. But I told you I had two examples of appreciations and things that I've seen in the literature. And the second comes from a book that I read. The book is called Spark: How Creativity Works, by Julie Burstein, who's actually here in the audience today. So we could ask her any questions that come about this book. And this is a wonderful book that was taken from a series of interviews that was done on a public radio program that Julie was the executive producer of, called Studio 360, where they were able to bring in all the most creative people really in the world and interview them about their creativity, about their lives. And what Julie did is she kind of distilled down what is it about them, what is it about their history that makes them creative. And just to give you a flavor for some of the people that she describes, there's artists, there's Broadway stars, there's TV stars, there's musicians, there's authors, there's playwrights. But the chapter that sparked my interest in just realizing how much neuroscience comes in to all the stories that kind of come across our desks is the first one on the list, Chuck Close. Chuck Close is one of the preeminent artists of our time. He's known as a photorealist. And I like to show this picture because it shows the expanse and the size of the art that he does. Here's somebody standing in front of his kind of prototypical pieces of art. It may look like a photograph. This is actually a painting. It's huge. It's 10 feet high by 7 feet wide. And he said that he likes to kind of put the viewer in Gulliver's Travel shoes, where he goes to the land of giants and you have to crawl over the crevices of this guy's nose and lips to kind of get into the art. So he creates these huge pieces. What's so interesting about Chuck Close is his history and how he ended up creating this kind of art. And it really comes from both his abilities and his disabilities growing up. So as he says, he had severe learning disabilities growing up. He had a hard time reading, he had a hard time doing math, he had a hard time writing. And teachers basically told him that, you know, you're just not very smart. And I just want to play a little audio clip here that gives you a feeling in his own words of what those early years were like.
I think I learned early on that since I also wasn't athletic and I couldn't run or catch a ball or throw a ball, that I needed to do something to keep people around me. So I began doing magic acts and puppet shows, and I began to realize that one of the things that I could do that my friends couldn't do was draw. And I was lucky enough to have the support of my family and to feel like I had something to say, even though I didn't say it in the traditional way. I was learning disabled, although in the 40s and 50s nobody knew from learning disabilities. I was just dumb.
Okay, so out of these learning disabilities, he certainly at an early age recognized his artistic abilities. At the age of 8, he was studying with a teacher, and he was drawing, you know, nude from nude models, which he says made him the envy of all the little boys in his neighborhood. He certainly recognized his artistic bent very early. But why faces? Well, that comes out of another issue that he had with his visual system. And here I'll quote from Julie's book. She says, he's also certain that his choice of subject, that is, the big faces, stems from another facet of his learning disability. He says, quote, I've had face blindness or prosopagnosia my whole life. Really, it's been a nightmare situation for me. Close once said, I don't know who anybody is and have essentially no memory at all for people in real space. But when I flatten them out in a photograph, I can commit that image to memory in a way. I have almost a kind of photographic memory for flat stuff. So his taking photographs of people, and all of his early work were self-portraits, but of his friends, were a way for him to actually commit those images to memory. Now, I've seen him interviewed in lots of, in a number of different situations. I highly recommend go look at the Stephen Colbert interview with Chuck Close. It's actually hilarious. But he says that, Colbert asked him, what does it look like when you're looking at me? And he says, well, every time your face moves, it looks like somebody different. So I have no idea that you're the same person. You and I can generalize across all the different angles. If I move this way and move this way, you don't think it's suddenly a different professor up here, it's still me. He is not like that. And he said that even friends that have lived with him for many years in the same house, he still doesn't recognize them. Now, of course, you can recognize the voice, but face recognition is hard. We're going to be talking about that. That is part of the visual system. Face recognition is a critical social aspect in our lives, and we know the brain area critical for that. It's in the temporal lobe, and it lights up when people are asked to recognize different faces. This is clearly gone in Chuck Close, but look at what he was able to do with the talents and the abilities that he had to create amazing art through his attempt of learning the faces in the way that he was able to. So let me just play the second and last quote, where he gives a really nice.
Also, part of my learning disability was being overwhelmed by the whole, and I found it to be particularly helpful to use a grid to isolate one small piece that I could work on and forget about the rest of the picture. But at a certain point in my work, I began to let the grid show and to leave it as part of the picture.
So what is he talking about, this grid? What he's saying is that, he not only flattened out the picture, but he actually subdivided it in different parts. Because what its process is, here is also a very famous big self-portrait from 1967, but what he did was the following. This is a photograph that he took of himself, and what he did is simply superimposed a grid on top of that, and then blew up that grid and painted each grid one by one. He was overwhelmed by the whole. He subdivided it and reproduced each little bit by bit, so he, as a person with his kind of brain, could understand it better. It turned out like this, and what he was saying is that at first he didn't let the grid show, but later, this is also a much more realistic one, later in other works, the grid became part of the work, and it was part of what he was able to show. Even working with this more confining grid, you can see as you look at his work all the different ways that he was able to use it, using different materials, sometimes spray paint, sometimes little dots. This is a stamp that he used to reproduce the same. This is Philip Glass, famous portraits of Philip Glass that Chuck Close is famous for. So prosopagnosia, part of the visual system that we're going to be talking about. The second aspect, I got to another part of the chapter where he talks about what he calls the event in his life. This happened in 1988, and this was a very rare spinal aneurysm that actually left him paraplegic. So he's in a wheelchair today. After a lot of rehabilitation, he was able to use his hands and still paint. So thank goodness, we didn't lose that gift. But the spinal aneurysm was severe. And we are also going to be talking about what is the organization of the spinal cord that removes both motor and sensory function at a particular given level. And so that is that aneurysm, which is a bursting of a blood vessel that basically killed the spinal cord at a certain level and removed all input. So all those axons going down to move your toe, if that spinal damage would eliminate that ability for you to move your toe or to feel the feedback from the sensory information from moving your toe. So what happened after the spinal aneurysm? He recovered, he was able to come back, but something very interesting happened, and that is a shift in his art. He moved from images like this and this to images like this. Color. Color came in, and many people have described these forms, these paintings, as much more emotional than the others. I don't know. I do think these kind of images where each one, you can see the grid, hopefully, and you can see that each one of these grids is not simply dots or spray paint, but complex designs, hot dog designs, ellipses, different shapes in different colors, that when you step back and see the whole, it's still a whole face. I think this is richer. I think this is, I think it is more emotional. But that's just my opinion. These are also very, very striking, especially I find this one very, very striking. But this is the same self-portrait. This is a later self-portrait, 1997. Very emotional. Why do I think it's emotional? Does anybody else have a feeling? Do they like the color ones? Let me show you a couple of other color ones. Same kind of techniques versus the black and white ones. What do you think the difference is? Do you like the black and white ones or the color ones? Do you see a difference kind of emotionally as you look at these pictures? This is not an art appreciation class, but it is a neuroscience class. And the way that you, the reason why I think that the color ones are more emotional is because that visual input is coming in, it's being processed by a structure called the amygdala. The amygdala is critical for both positive and negative emotion. The amygdala is supercharged in people that suffer from PTSD. In those people, even the most innocuous stimuli come in and trigger huge emotional negative memories from whatever trauma they experienced. Why? Because the amygdala is a structure that gets inputs from all sensory modalities and rich inputs from sensory modalities, but its output is to the areas that make your heart beat faster and make your stomach get all in knots when you're nervous, or calm the stomach down. So the amygdala has this interesting place in the highways of the brain between sensory input and emotional output. And that's what's causing some people to think this or this is more emotional or not, or maybe you think they're both the same. Okay. And lastly, the last element of this chapter that I thought was so interesting relative to neuroscience is that, in fact, a neuroscience paper in the journal Science, which is the best journal in science, was written. It was written by a colleague right here in the psychology department at NYU, Dennis Pelley. And what he looked at was the visual perception, comparing this. Now, it's very hard. I've blown this up. Can you see a face here?
Yes.
Yes. Can you see it better there?
Yes.
Yes. Okay. So what happens is when the pictures are blown up so big, and so I'm simulating you at a museum looking really close up, and I can't see any of the contours, and moving back, moving back, moving back, until you see that face pop out. What makes that face pop out? And the whole paper was about kind of the visual illusion that Chuck Close has created in his art. And it's actually brought out something that people didn't realize before. That is the size, in this case, the size of his grids affects the shape and the contours that you're able to see. At a certain distance, the nose here pops out. But if you're too close, it's just a flat surface. And the whole paper was about what about the different elements were making that nose pop out. And the critical element was size. And so the research for this paper was going to the museum and going close and going far away and going close and going far away and just doing the kind of self study on what you see at different levels. And it's ironic for a kid who couldn't do math at all to then have his work studied for these very kind of technical and mathematically oriented principles of how the visual system works. In fact, I don't know, it must have been about 10 years ago, Chuck Close came here for a seminar on art and neuroscience. And this paper had just come out, and it was discussed extensively. So that was very exciting. But just to give you a feeling for all the different ways, even you might think, I'm reading this book for pleasure. So many different aspects of neuroscience that I saw that we're going to be covering, so I wanted to share that with you. Okay, so I'm going to end with a quote at the end of this chapter, and it's a quote by Chuck Close, and he says, "Inspiration is for amateurs, and the rest of us just show up and get to work. But so much of it comes out of the process. If you try to preconceive everything you do and conceptualize it, you're going to do the same thing over and over. If, however, you get busy and things occur to you in the process, you make the rules, and therefore you can break them." So he's all about kind of spontaneity, even in his kind of grid-like work. But despite the fact that he thinks inspiration is for amateurs, I think that many creative people use inspiration a lot. I certainly use inspiration in my work as a scientist. And again, this is what I want you guys to be focused on as we go through this class. So Monday, come back. We'll start with neuroanatomy. Anybody interested in any of the programs that I talked about, come up, sign up for the email account, and I will see you on Monday.